Hello everyone, welcome back to Biogenesis. Today our topic is cell cycle regulation. What does it mean? Cell cycle regulation means there are some proteins that control the mechanism of cell cycle checkpoints, and by this way cell cycle progression is regulated. So, progression through these checkpoints is largely determined by the activation of two kinds of regulatory proteins, cyclins and cyclin dependent kinases. What are cyclins? Cyclins are regulatory proteins. They bind to their specific CDK, that means cyclin dependent kinases, at different stages of cell cycle. There are mainly four types of cyclins that are activated in different stages of cell cycle and regulate the cell cycle mechanism. Those are cyclin D, E, A and B. Next is cyclin dependent kinases. It is a family of serine or threonine protein kinases. Serine or threonine protein kinases are actually enzymes that catalyze the transfer of a phosphate group from ATP, adenosine triphosphate, to a protein substrate. More precisely on a serine or threonine amino acid residue. That means every protein is made up of different type of amino acid, and this serine or threonine protein kinase actually acts by transfer of phosphate group from ATP to particularly serine or threonine residue amino acid, serine or threonine amino acids of the protein. And the cyclin and CDK complexes are formed, and the activity of cyclin CDK complexes is modulated by phosphorylation and dephosphorylation events. Sometimes phosphorylation can activate, and sometimes dephosphorylation, that is removal of phosphate group, can activate different events. There are different types of cyclin dependent kinases: CDK4, CDK6, CDK2 and CDK1. Next is tumor suppressor proteins. Tumor suppressor proteins are a type of proteins that can halt the cell cycle to prevent uncontrolled cell division and thereby preventing the development of cancer. If the cell is not prepared well, that means if there is any DNA damage in the cell, then these tumor suppressor proteins are activated and started their regulation program. First one is P53, which is known as guardian of the genome. It is a multifunctional protein that activates in response to DNA damage, and it can halt the cell cycle. It can halt the cell cycle if any DNA damage is there and allow DNA repair by increasing the level of P53 protein. If there is no chance for DNA damage repair, in case apoptosis takes place. In response to DNA damage in mammalian cell arrest at G1 phase by this P53 protein. How it actually works? P53 activates another protein, that is P21, and then P21 acts as CKI. What is CKI? Cyclin dependent kinase inhibitor. So, cyclin dependent kinase inhibitor then inhibits CDK, cyclin dependent kinase activity, and blocks G1 to S transition. If in G1 phase there is any DNA damage in cell, then the cell needs to be stop their cell cycle progression and it is allowed to repair the DNA damage. Next one is retinoblastoma or RB. Retinoblastoma actually prevents tumor formation, that means uncontrolled cell growth, by repressing transcription process. Okay. By this process, it regulates transition from G1 to S phase by controlling the activity of a transcription factor known as E2F. E2F is a transcription factor. It helps in transcription process. So, retinoblastoma actually controls the activity of E2F and thereby regulate the transition from G1 to S phase. When retinoblastoma binds to E2F, it is a transcription factor. It is in active state, and in this case E2F cannot bind to DNA and transcription blocked. If retinoblastoma is phosphorylated at its different residue, then it becomes inactive. E2F is released from retinoblastoma because retinoblastoma still cannot binds E2F. Then E2F easily binds to DNA and different cell cycle protein expression or different cell cycle protein formation takes place, and easily the cell can progress from G1 phase to next phase. Okay. If P53 protein, because P53 protein can control the cell cycle progression, so if P53 protein is mutated, then it lost its function, and as a result arrest at G1 phase is prevented even in response to DNA damage. If DNA damage is already there but P53 is not activate its function. Okay. And this condition is found in different human cancers. Another tumor suppressor protein is P27. P27 also binds to cyclin CDK complex and prevent also G1 to S transition. Reduced levels causes uncontrolled cell growth and cancer. If P27 level is reduced, then cell can growth in uncontrolled manner and cancer will happen. In G1 to S transition, when cell complete its different events in G1 phase and it is ready to enter next S phase, then different cyclins and CDK proteins help the transition process. Okay. In G1, cyclin D acts as early cyclin and cyclin E acts as a late cyclin by binds to their respective CDK. Like cyclin D binds to CDK4 or 6 and cyclin E binds to CDK2. When growth factors binds to cell surface receptors, then different pathways like RAS, RAF and MEK kinase pathways is activated. By this pathway, RAS, RAF and MEK kinase pathway, cyclin D regulatory protein is formed. Then cyclin D binds to its respective CDK4 and 6 kinase proteins. Then cyclin D CDK4 6 binds to active retinoblastoma, and in this condition retinoblastoma actually binds to E2F transcription factor and then blocks E2F transcription factor and transcription is blocked. We already seen in the previous slide. So, when retinoblastoma binds to E2F transcription factor, in this case E2F cannot start any transcription process and retinoblastoma is in active state. But when cyclin D CDK4 complex is formed, it phosphorylates retinoblastoma in different residues and retinoblastoma becomes inactive. By this process, E2F is easily released from retinoblastoma and E2F is activated, then it binds to DNA and S phase gene transcription and as well as S phase different cyclins are synthesized. That is how G1 to S transition is occurred. What is retinoblastoma? Retinoblastoma was first identified as the product of a gene that is mutated in retinoblastoma disease. It is a rare inherited childhood eye tumor. If functional retinoblastoma is absent, then it not only cause retinoblastoma, that means it not only cause eye tumor, but it also causes different type of human cancers. Also, in this case, there are different oncogene proteins. What is oncogene? Oncogene is a gene that is responsible for cancer. So, oncogene proteins here is RAS and cyclin D. So, RAS and cyclin D helps in cell proliferation process. That means it helps in cell proliferation and as well as cell division, and cell can divide rapidly, and this will cause sometimes cancer. So, when there is any default in any cell, this retinoblastoma protein actually blocks the cell cycle progression. Also P53 protein, P27 protein blocks the cell cycle progression to control the uncontrolled cell division, and that is how prevents cancer cell formation. Okay. When the cell is allowed to enter the S phase, that means when cell enter G1 to S phase, different mechanisms takes place in S phase. Most importantly, DNA replication. Okay. In S phase, cyclin E or cyclin A protein is activated, and cyclin E or A protein binds to its respective CDK2 and continue the process. In S phase, cyclin E and CDK2 complex is formed and it initiates S phase by activating DNA synthesis at its replication origin. Then cyclin E CDK2 activates the MCM helicase protein. What is helicase protein? Helicase protein is a protein that actually unwinds DNA. Normally DNA is in double stranded. When DNA replication is started, MCM helicase protein comes and it unwinds DNA, and the DNA strands are separated from each other at the replication origin. Suppose this is the replication origin point. So, cyclin E CDK2 activates this MCM helicase protein by phosphorylation process and initiate DNA replication. Then the MCM protein moves away from the origin with the replication fork. Okay. By this process, when the MCM protein moves away from the origin with the replication fork, it unwinds DNA and replication process is going on. When high activity of CDKs during S, G2 and M phases occurs, it prevents the MCM proteins from reassociating with replication origin. Next, the amount of cyclin A CDK2 complexes increases because in S phase cyclin E and cyclin A, two types of cyclins are there. At first, cyclin E CDK2 actually starts the replication process. When replication process is already started and it is going on, then the amount of next cyclin protein, that is cyclin A CDK2 complex, increases. It ensures that in a cell, DNA is replicated only once, not twice. DNA is replicated only once per cell cycle. So, in this case, cyclin ACDK2 prevent the assembly of other replication complexes. So, MCM helicase proteins, when unwinds the DNA, it's move away along the replication fork and starts the replication process. Then cyclin ACDK2 complex amount increases. That means it does not allow any additional replication complexes to recruit at the same origin and again start the replication process, because per cell cycle in per cell cycle only once DNA is replicated. Then the cell complete their cell cycle and S phase to G2 transition takes place. Here is another protein, that is SYK1. SYK1 is actually acts as a CDK inhibitor, cyclin dependent kinase inhibitor. It actually inhibits the cyclin dependent kinase activity, mainly S phase cyclin CDK complex activity. So, when SYK1 is phosphorylated by SCF complex, what is SCF? SCF is a complex protein, SKP1, Qlin and F-box proteins together form the SCF complex. So, SCF complex phosphorylates SYK1 and targets it for ubiquitination and proteasomal degradation, and that is how SYK1 protein is destroyed, and easily cyclin CDK complexes activity is restored and it starts DNA replication process, and DNA replication easily start, and the cell then completes the different events in S phase, and then from S phase they enter into the G2 phase. When cell completes DNA replication and enter into G2, and then from G2 phase cell enter into M phase by an interesting control mechanism. In M phase, cyclin B is working and it binds to its respective CDK1 and form complex. So, cyclin B binds with CDK1 at the end of G2 phase and form MPF, that is mitosis promoting factor. So, then when MPF, mitosis promoting factor, is formed, CDK1 is phosphorylated by a protein kinase, that is CAK, CDK activating kinase. So, by this name we already easily understand that CAK actually activate CDK kinase protein. So, CAK phosphorylates the CDK subunit of MPF at threonine amino acid 161 residue. Another protein kinase WEE1. WEE1 kinase has a dual phosphorylation mode. That means it phosphorylates CDK1 subunit of MPF at two positions. One is tyrosine 15 and another one is threonine 14, and that is how it inactive cyclin B CDK1 complex as well as MPF complex. When cells are ready to enter from G2 to M phase, a dual specificity phosphatase named CDC25, it is a phosphatase, CDC25 is recruited, and dephosphorylation of tyrosine 15 and threonine 14 actually occurs. But active phosphorylation at threonine 161, that is caused by CAK protein. CAK protein phosphorylates CDK1 subunit at threonine 161 residues. So this active phosphorylation at threonine 161 is still working, and then MPF becomes active and cell enter mitosis. When cells reach appropriate size, that means cells have enough resources, enough nutrients, enough proteins, RNA, then another component named CDR2, it inhibits We1 kinase proteins and allowing CDC25 to activate mitotic CDK, that means CDK1. That's how cell from its G2 phase easily enter into M phase. Next, the cell enter prophase of M phase, and after prophase it enters metaphase, where different protein complexes, in metaphase different protein complexes that together establish the linkage between sister chromatids are called cohesins proteins. When chromosomes are in equatorial plane, in metaphase stage, when chromosomes are in equatorial plane, gradually the spindle fiber apply forces to pull the two kinetochores toward the opposite spindle poles. But sister chromatids do not separate. We all know that two sister in metaphase stage, the two sister chromatids are seen in equatorial plane, and the two kinetochores in different sister chromatids bind to the spindle fibers. But in this case, when the mitotic spindle is in state of tension, that means it apply force to pull the two kinetochores toward the opposite poles, it try to separate the sister chromatids, but sister chromatids do not separate. So, to separate these sister chromatids, cohesins protein should be removed. These cohesin proteins should be removed because it link between sister chromatids. It establishes linkage between sister chromatids. Let's see a diagram. This is the cohesin protein. It is a protein complex because different type of protein make complex and form cohesin proteins, and it establishes the linkage between sister chromatids. Okay. A protease known as separase. Separase is a protease. See the diagram. It is the separase. It is a protease that cleaves the cohesin protein subunit ACC1. It is a subunit of cohesin protein. This separase cleaves the ACC1 subunit and then breaks the protein circles, and by this process sister chromatid can easily separate. At the onset of anaphase, that means when metaphase is about to end, APCC, it is a complex, APCC is directed by CDC20. APCC and CDC20 complex is recruited and it ubiquitinylate securin. It ubiquitinylate securin. That means it try to destroy the securin protein, which is subsequently degraded by proteasomes, and this degradation process activates the separase proteins. Then the separase protein is activated and then it cleaves the SCC1 subunit, allowing sister chromatids to be pulled apart by the spindle apparatus, and then it moves towards the opposite poles. By this process, the two sister chromatids are moved to the opposite poles, then anaphase is completed, then the cell enter telophase stage, and then finally karyokinesis process is completed. After this process, the cell enters cytokinesis, and in cytokinesis process, after chromosome segregation, the two daughter cells are produced and cell division is completed. Okay. So, if you have not watched the previous videos that is related to this topic, then please watch them. It will really help you to understand this topic better. Okay. Thank you everyone.